In this second screencast for Chapter 13, we will cover the specific, or also called the adaptive immune response. The specific immune response protects your body from pathogens by producing cells and products designed to fight one specific pathogen. There are two complementary components to the specific immune response. These are the antibody-mediated and the cell-mediated responses. Let's start with a cast of characters. Remember, a pathogen is a disease-causing agent. On the surface of the pathogen are identifying molecules different from the markers present on your cells. These identifying molecules are collectively called antigens. Antibodies are Y-shaped proteins that attach to the antigens on a pathogen. The place on an antibody that attaches to the antigen is called the antigen binding site. The three-dimensional shape of the antigen binding site determines which antigen, and therefore which pathogen, the antibody will be effective against. Antibodies are produced by B cells. Each B cell produces one specific antibody. Plasma cells are clones of B cells that produce an antibody needed to fight an infection. Plasma cells release large quantities of the antibody into the bloodstream. Macrophages destroy pathogens by eating them. Like all your body cells, macrophages display MHC markers which identify them as belonging to you. Helper T cells are the generals in the immune system army. They produce chemical signals that coordinate the immune response and signal the production of more white blood cells. It is the production of memory cells that provides long-lasting immunity. They are produced during the initial infection but are held in reserve instead of playing an active role in the initial infection. Now let's look at how all these cells work together during the antibody-mediated response. A pathogen has gained access to your body. Macrophages, which constantly patrol your body, find one of the pathogens and realize the antigen does not match your MHC markers. The macrophage responds by eating the pathogen and digesting it. Kind of like a badge of honor, the macrophage displays the pathogen's antigen on its plasma membrane. The macrophage next communicates with the helper T cell. The helper T cell recognizes the macrophage as one of the good guys and responds to the antigen by producing chemical signals which cause your body to start randomly producing a variety of B cells, each with their own unique antibody. When a B cell is produced, which has the antibody, whose antigen binding site can grab on to the antigen of the pathogen, that B cell becomes a sensitized B cell. The sensitized B cell communicates with a helper T cell. The helper T cell now produces chemical signals which cause the cloning of that sensitized B cell. These clones are called plasma cells because they produce large quantities of the antibody, which is then released into the plasma. Some of these plasma cells are held in reserve as memory cells. This process of producing a variety of B cells and then cloning the B cell which produces the needed antibody is called the clonal selection theory. The needed antibody is transported throughout your body, helping to eliminate any pathogens that are present in the blood and also in the extracellular spaces between the cells. Some pathogens, viruses for example, must gain access into your body's cells. Once inside your cells, these pathogens are hidden from the antibodies. Another immune response is needed to eliminate these infected cells. That other response is the cell-mediated response. Its job is to eliminate the infected cells. As in the antibody-mediated response, macrophages first find the pathogen which has invaded your body. The macrophage eats the pathogen and displays its antigen on the plasma membrane. The macrophage communicates with the helper T cell. The helper T cell this time produces chemical signals that cause your body to start producing lots of different T cells. These T cells can kill infected body cells by producing a chemical which eats holes in the plasma membrane, therefore killing the infected cell. The T cell, which is going to be effective in fighting the infection, is identified by communicating with the macrophage, which displays the antigen. This particular T cell is cloned. The clones are called cytotoxic T cells. Most of these cytotoxic T cells immediately join in the battle, but some are held in reserve as memory cells. The cytotoxic T cells kill the infected body cells, and now the pathogen has no place to hide, and the pathogen is quickly eliminated from your body. The production of memory B and T cells gives you active immunity to the pathogen. These memory cells typically stay with you for the rest of your life. If the pathogen gains access to your body again, you don't have to go through all the steps of the antibody and cell-mediated response. Instead, the memory cells immediately spring into action and eliminate the pathogen before you get sick. Active immunity can be the result of catching a disease and successfully fighting it off, or by receiving a vaccine. A vaccine can be made of dead or weakened pathogens, pieces of pathogens, or some other product produced by the pathogen. Your body responds to the vaccine just like the real pathogen. You go through all the steps of the specific immune response and produce memory B and T cells. The vaccine gives you active immunity safely without having to suffer through the disease. Let's stop here for the second screencast.